Hi, I am Nur Farsha Yasmin binti Ahmad Fauzi. Today, I will help you to discover about breast cancer. Breast cancer is a disease in which cells in the breast grow out of control. There are different kinds of breast cancer. The kind of breast cancer depends on which cells in the breast turn into cancer. The breast cancer will grow out of control by its stages. Breast cancer staging describes how far the cancer has spread within the breast and other parts of the body.
Starting with stage 0, breast cancer is known as broad-invasive breast cancer. Abnormal cells are present but they are contained inside the milk duct in the case of ductal carcinoma in situ DCIS or global in the case of lobular carcinoma in situ LCIS. Next, stage 1 breast cancer is the earliest stage of invasive breast cancer. Cancer cells have spread from the original location in the ducts or lobules to the surrounding breast tissue. The tumor is less than 2 cm in size. In stage 2, invasive breast cancer, which tumor size varies from less than 2 cm to greater than 5 cm. In stage 3, tumor size may vary from less than 2 cm to greater than 5 cm. It has spread to several lymph nodes. Lastly, in stage 4, known as advanced breast cancer, which has spread to other parts of the body such as the liver or bones. Next, what is a microarray technique? DNA microarray is a technology which enables the researchers to investigate issues which were once thought to be non-traceable. One can analyze the expression of many genes in a single reaction quickly and in efficient manner. DNA microarray technology has empowered the scientific community to understand the fundamental aspects underlying the growth and development of life, as well as to explore the genetic causes of anomalies occurring in the functioning of the human body. Thousands of spot samples known as probes, known identity, are immobilized on microscope glass slides or silicon chips. The spots can be DNA, cDNA, or oligonucleotides. These are used to determine complementary binding of the unknown sequence, thus allowing parallel analysis for gene expression and gene discovery. There are 3 types of microarrays, which are microarray expression analysis, microarray fermentation analysis, and comparative genomic hybridization. In detecting breast cancer, microarray expression analysis is the most suitable. For this analysis, the cDNA derived from the mRNA of non-genes is immobilized. The sample has genes from both the normal as well as the diseased tissues. Spots with more intensity are obtained for diseased tissue genes if the genes is overexpressed in the diseased condition. This expression pattern is then compared to the expression pattern of a gene responsible for a disease.
Next, I, Zawati, will carry on to explain the methods used in the experiment. Firstly, we will look at how the researchers managed to turn smartphones into cancer detecting. The device is called fluorescent. Fluorescent is a portable fluorescent microarray imaging system seamlessly connected to a smartphone. The smartphone acts as the central component of the detection system, capturing images of the microarray spots using its built-in camera. The fluorescent system operates similarly to a fluorescent microscope, utilizing 2 light spectrum filters, one for exciting the fluorescent dye and the other for capturing the emission spectrum. By analyzing the captured image, using specialized software, we can identify the expression of the BRCA-1 gene with accuracy and precision. But before we get ahead of ourselves, what is BRCA-1 gene? Put it simply, BRCA-1 gene is a tumor-suppressing gene. This gene is involved in repairing damaged DNA. It is located in the nucleus of many types of normal cells. The mutation of this gene increases risk for cancer. In short, the BRCA-1 gene interacts with several other proteins to mend breaks in DNA. Now, this break may be caused by natural radiation or during chromosomal exchange, genetic nutrient preparation for cell division. Let's briefly talk about the materials and chemicals used in the study. The researchers obtained DNA oligonucleotide for exon 11 of the BRCA-1 gene conjugated with cyanide-3 from sigma aldrich. Reagents like bovine serum albumin, sodium dodecyl sulfate, and saline sodium citrate were utilized as a sieve. They also used phosphate buffer silane, white membrane filter paper, and ionized water from the liquid system. The study employed paper microfluidics for pressure-driven fluid flow. They designed channels using AutoCAD and printed them using a wax printer. The printed channels were then baked to spread the molten wax in NC filter paper. They then evaluated the fluid flow before and after baking and examined the reagents using scanning electron micrographs. This helped them optimize the channel determinations for the DNA microarray experiment.
Alright, next, let's take a look at the fluorescent device. It's for capturing and analyzing fluorescent signals from the DNA microarray. The fluorescent device is specifically designed for detecting cancer biomarkers and features excitation and emission filters that gathers, collimate, and disperse light within the 520 to 575 nanometer wavelength. The green LED light source is used to project light through the microarray, while a smartphone placed on top to capture the emitted fluorescent signals. To ensure accurate readings, the entire device is printed black to block external light and minimize diffraction. Since the sensitivity of the device for detecting the BRCA1 gene, the limit of detection or LOD are also being implemented. Using arrays containing CY3 DNA oligonucleotide probes at various concentrations ranging from 0 to 10 micrometer. The arrays were prepared by serial dilution with 3 times SSC buffer, and a control background array consists of the buffer alone. Capturing image of the arrays and analyze the fluorescence intensities allows to calculate their LOD. This evaluation highlights the device sensitivity in detecting of the BRCA1 gene, a crucial parameter for its performance assessment. In the effort to detect the BRCA1 gene, a crucial DNA hybridization assay is being used. This assay involves the use of short oligonucleotide primers designed specifically for exon 11 of the BRCA1 gene, served as a probe DNA. Carefully performed on the paper microfluidic device, the assay consists of 4 main steps: loading, blocking, loading, and washing. By utilizing the specific primers and analyze the hybridization result, high specificity results were achieved in detecting the BRCA1 gene. Additionally, the negative controls are being incorporated as to ensure an accurate and reliable results. As for the recaps, the smartphone-based DNA microarray analyzed using paper microfluidics and the fluorescence setup has demonstrated great potential in detecting BRCA1 gene associated with breast cancer. This portable and cost-effective system offers a promising approach for a point-of-care application, especially in regions with limited access to advanced laboratory equipment. Early detection of the BRCA1 gene can have a significant impact on breast cancer diagnosis and treatment outcomes.
The ultimate outcome from this method has 3 viewpoints, including a paper microfluidic smartphone readout output and DNA hybridization assay.
According to the result which should detect the breast cancer gene, this approach can resolve 23.7 nanometer line pair for the blue highlight region in Figure A. Figure C and Figure D illustrate the cross-sectional intensity profile and the full white half maxima that was taken from Figure C respectively. The region with orange highlight was found to have a resolution of 32.1 nanometer. Figure H displays the full white half maxima derived from Figure G cross-sectional intensity profile. The result highlight using a 1951 USAF resolving power test target. This printing and baking technology demonstrated breast cancer gene resolution. After baking, in Figure B display 20mm square blue dot patch of the test target. The resolution target cross-sectional profile shown as the blue line in Figure B. 50mm square area with orange dot after baking. A cross-sectional profile of the orange line resolution goal in Figure. Based on the figure, it can be summarized that the peak was discovered to be at 260 nanometer and the DNA absorbent with concentration up to 0.6 nanogram. The absorbent method of detection is accurate in Figure 6D. Fluorescence spectroscopy was found to have a detection limit that is 10 times lower that of fluorescent imaging-based quantification in Figure 6C. The ratio of the amount of DNA to dye which was calculated using the dye combined absorbent and fluorescent emission data in Figure 6C, E and 7 was determined to be 8.55 DNA per dye. This result illustrates starting copy number for the 10mm working solution and 1 liter working volume employed in assay was discovered to be 2.85 times 1013 DNA copies. It can be determined minimum of 11 log 10 DNA copies which correspond to 0.4 mole load with dilation factor of 250 are need to identify breast cancer gene using the paper microfluidic smartphone readout system.
In conclusion, the simultaneous examination of the expression thousands of genes in tissues, organs, or cells can be accomplished with the help of microarray technology. Microarray technology is a flexible method that may be used in a wide range of techniques to better understand how cancer develops, to manage and treat patients, and to detect people who are prone to get cancer. A portable optical sensing technology called fluorescent was created and tested for the purpose of finding breast cancer biomarkers related to the BRCA-1 gene. It has a detection sensitivity of DNA copies and can resolve 23.7 meter line pairs. It is affordable, simple to use, and expandable. That's all from us. Thank you.